I put in the Slack channel the link to the tutorial that we're going to do. But I'm going to show you where you can find much more tutorials. So if you go to bonvinlab.org, up there, I don't know if it's better visible or not. That's the group, the web page of my group. And in that page, there is the education link. If you click there, you will find a lot of different things. This one, by the way, is the structural bioinformatics and modeling. It's a master course I'm giving, and we have an extensive tutorial or practical part, which starts with homology modeling, using Swiss model in that case. We are running molecular dynamic simulation of a peptide. This is using GROMACS, and we're using NMR box as a system for that, which are virtual machines in the US that are freely accessible. And then we do docking with HADDOCK, where we use the conformation of the peptides from the MD to do docking with the server. Okay, so that's quite expensive. So next to all the tutorials that you have heard about MD, that's yet another tutorial based on GROMACS, that one. There are a number of lectures here that have been recorded from previous meetings and summer schools. And then you have different links. There's a best practice guide where there is the to-dos and don'ts for HADDOCK. So it's telling you how to prepare structures and things like that. So if you're going to do some HADDOCK modeling, you might want to look at those. There's a website to generate restraints. We have tutorial for version 2.4 of HADDOCK, which is the version running on the web portal. So here you will find a lot of tutorials for HADDOCK 2.4. So basic web server tutorials, if you want to do those. So there is an antibody antigen docking using the web server. There's a glycan tutorial using the web server. And then there are more intermediate ones. Some of those do require Linux access. Some of those are purely web-based. Cross-linking tutorial, antibody antigen, more command line. That's also a funny one. Using another tool that we developed this week, and then we have more advanced tutorials, some of them for small molecule docking. So you can find different things here. And you find also documentations. So this is the old. And then you find tutorials, so we have different things also, but you find the HADDOCK free tutorials, and this, it's something, so we are porting slowly our tutorials to HADDOCK free. So we have the antibody antigen one, which is what we're going to do today. We have another body antigen one, which is related, protein glycan modeling, HADDOCK free. These are all command line tutorials, basically. And we also have now a protein DNA docking and peptide dockings. And more advanced one. And all at the bottom, you will find also the user manual for HADDOCK free. A tool to generate restraints for HADDOCK, which is an online server. And then there's a software documentation for HADDOCK free, which is more for the software developers. Okay, so the one for which I put the link in the Slack channel is this antibody modeling tutorial. And it has different steps. So we're going to go through it together, or at least you can go on your own, but I will also demonstrate things. As you can see here, there is a setup to do that on Fugaku. So HADDOCK free has been installed, the data are there, so you could run on Fugaku, do all the command line work. But for the purpose of this tutorial, I suggest if you have a Gmail account, we can use Colab resources from Gmail, because that's much user-friendlier to run the tutorial. You can reuse it at any time, even when you not have access to Fugaku. If you don't have Gmail account, then you need to stick to the Fugaku. But in principle, the tutorial in the Jupyter notebook running on Colab does the same thing as what you will be doing command line in Fugaku. And actually, it runs a bit faster on Colab. So what do we have here? So there is a a general introduction. Actually, I've been running the command line version of this tutorial in another school that we run in the context of Bar Excel, and this one was in Sofia, and this was recorded. So there you can see all the command line work associated with this tutorial. So if you want to get more details. So by now you know what an antibody is. Okay, so we're going to model the binding regions, and this is where the loops are. In this tutorial, so if you are following this for the Google Colab, it's not relevant, but just the philosophy of all our tutorials are rather similar. So we have color coding. So orange will be a question, so you should think about it, okay? If you see blue, it's an instruction, so we need to do something. Green is usually used for visualization. So we use PyMol a lot, but in some of our tutorial, it's camera used. So these are commands to visualize things. And black, it's something that you need to do in a terminal, okay? So you need a command line interface. So you could run things on Fugaku for that. You can also, if you have a Windows subsystem installed on your Windows laptop, then you can run things in the Windows subsystem, and you can install HADDOCK there. If you have a Mac, you can run simply on the terminal of your Mac and run things there as well. So since we are not doing heavy computing or a huge number of computations, we don't need per se heavy computing power. So this is what I've been explaining with all the modules. And so they are, if you want to redo things on your own, you can, on your own computer at some point, you can follow those instructions, download the data, so the data are available, so they are not limited. They are not only on Fugaku, but you can always access it. And this is the instructions that are specific for Fugaku. So if you don't have a Gmail account and you don't want to use Colab, you will have to either create a terminal session to Fugaku or use on demand and open a terminal session and follow the instructions, okay? So there are specific instructions. So go into your data directory, create a direct, and then unzip the data that are placed on Fugaku. So if you don't have Gmail, this is what you will need to do in order to be able to run things. You start an interactive session on a node, and then once you are in the node, you can start executing the tutorial, basically. We could also submit the HADDOCK calculation itself to the batch system and run things, but you can also run interactively. If you run this tutorial on Fugaku using a full node, I think it's 48 cores, the HADDOCK workflow will complete in about 25 minutes. If we use the Colab resources, it should take 11 minutes. So now if you are here, there is this Colab notebook link. Okay, so if you find that link, and I think I also put the link directly in a Slack channel, so we can click on that link. And now, if you look at the URL, basically the link is calling Google Colab, and it's getting the notebook from the GitHub repository of HADDOCK 3. And the notebook has the similar information as the website that was showing you. Okay, so now I'm not yet connected to resources. I'm logging into Google because you see this icon, so that's me. By the way, you know who this person here up there? It's very small, the icon, but you know who that is? Captain Haddock, yes. Okay, so the software is not called after a fish. No, it's called after Captain Haddock, who is the good friend of Tintin. So I'm logged in. So if you want to use Colab, you need to log in with your Gmail account. And if you are logged in, you need to connect to a free resource, cannot connect to TPU backend, usage limits, okay. Then I need to connect without a TPU, or maybe I should try again. Maybe too many of you connected already, I don't know. Let's try, sometimes we might not. Close. So then we need to change the run mode. Are some of you connected already? Okay, yeah, so maybe that's, so the TPU is kind of, it's nice because it's giving you more cores. If you connect to a CPU, I think it gives you more 4 cores. So that means that things are going to run slower. It's not that important for the tutorial because when we go to the analysis part, we are using pre-calculated data that are downloaded in the notebook. So I'm going to try once more, and if it kicks me out, okay, close. So then I connect to a CPU. Stop. We don't need GPUs. Change run type, so I'm going to go the CPU one. Save, connect. Yes, okay. So if you have TPUs, you're in better shape than I am. Okay, so now I'm connected. Who has used Colab notebooks before? Okay, so you know it works. So there are, of course, there are text. So this is the same text pretty much as you see on the website of the tutorial that we started with in a notebook. By the way, what is rather new, before there were hacks for that, but the notebook also gives you access to a terminal session now. So you can have a command line session as well, and you can explore what's happening, so that's quite nice. So this is the explanation about HADDOCK 3, but you know everything about that. And then you start getting the cells here. So the first thing that we're going to do is to install HADDOCK 3, and then we need BioPython and Py3DMOL for visualization. So you just click on that cell, run anyway, and now it starts downloading HADDOCK 3 and it's going to install it. So if you want to know what's happening here, you can unfold this. And basically, these are the 3 commands that are being done to install HADDOCK 3. So it's doing a pip install HADDOCK 3, and we are installing a specific version because this is the version that the tutorial was tested on. So that to make sure that we don't, your future version of HADDOCK might have changed and then you don't get the same thing. So we say we run the version with which the tutorial was created. We install BioPython and we install Py3DMOL for visualization in the tutorial. I was trying to run that tutorial, by the way, on Fugaku using on-demand because there is a Jupyter interface, but there are some issues with the executable that's needed by HADDOCK, and I couldn't solve them before the course. The local installation, the one for which the instructions are on the website, this is a fully functional version on Fugaku, so that's working. But if you do this pip install in a Jupyter notebook on Fugaku, there are some issues there. Okay, so the installation is done. We have installed HADDOCK 3. We have a working version. We are ready to go. So now we're going to download the tutorial data. So this is basically now downloading the data from that are available again from the website. So you see this served drive, and it's going to unpack things there. If I clicked on this icon, then I can see what is the content of my machine, and you see that I have this directory here, which was just created. It was not there before. And you see in this directory now we have, so these are the data that we downloaded. They are PDB files, so these are the input files that we need to set up the docking. There's a restraints directory. This contains the restraints for HADDOCK to define the docking. We have runs. These are pre-calculated runs so that we can analyze results without having to wait for the data. We have scripts and we have workflows. These are the workflows to give to HADDOCK. So in principle, everything is there, even the data that have been calculated beforehand, so that you don't have to wait for the computation to finish to be able to proceed. And if you are curious, you can also open a terminal session, and then you can give here Linux commands and see what is there. So you can see the same thing. So that's quite nice as well. Okay, so we have the data. So we have these different directories that are there. So now we are going to prepare the files for docking. Okay, so we're going to prepare things to do docking here. We're going to download the files from the PDB directly. The files are pre-processed, they are present, but just to show you. And for this pre-processing of files, we are going to use a software set called PDB Tools, which is also available as a web service on our site. So it's a collection of Python scripts, actually, that I use to message PDB file. Okay, so, and you can use them at the command line level, and you can pipe one command into the other. So what we're going to do, so antibodies consist of two chains. Okay, so we have heavy chain, light chain. For HADDOCK purposes, one molecule should be one chain. Okay, so we need to process the antibody to make it such that it's only one chain in terms of chain ID, A, for example. But you also have to make sure that there is no overlap in numbering between the two chains. Otherwise, if you have two residues that have the same numbering, then something will go wrong. So we're going to fetch first the file from the PDB. We're going to clean it a little bit to make sure that it has proper format. We select a specific chain. We remove heteroatoms. This is not, in this case, we just do it, but this is not something that you want to do blindly. But if you download a structure from the PDB, a crystal structure, do realize that in most cases, there will be crystal water in there. There might be small molecule co-crystallized with your protein because they were in the crystallization buffer. So there is junk, basically, in the coordinate files. And if you use it straight, without looking at what's in there, if there is water in your binding site or there are some other things, a phosphate ion is there, it might simply block your binding site. So look at what is in there. If you're using AlphaFold model, look at the models. There will be a lot of spaghetti in many cases. Remove the spaghetti, okay? Because these are disordered regions and they will only hurt you. So if there is disordered regions, don't try to model those. Better remove those things than use them in the docking. So never use blindly a file without looking at what's in there. The other thing with antibodies, and that's a bit the nasty thing of antibodies, is that there might be insertion. So in the loop, the CDR loops of antibodies, there is a numbering where people, you will see, if you open the PDF file in the text file, you might see there's residue 100, and then you see residue 100A, 100B, 100C. And if you have those, most software, I think GROMACS will probably have similar problems, will only read one of those because it reads 4 times residue 100 and it uses the last one most likely. So we need to renumber those. It means shifting the numbering of your sequence so that there's a unique numbering of those residues. So this is very specific to antibody. So this fixed insertion does that. The other problem that you might have with crystal structure is that, especially if you have high resolution structure, a side chain might exist in two conformations. Meaning in a crystal structure, they see two conformations of a side chain. Maybe occupancy is 50%, so 50% is one, 50% is the other. So your coordinate file has a duplicated side chain. And then what do you do with that? Okay, one amino acid has only one side chain, so you have to do something with that. So you might select, so if you use this select, it's basically select alternate location. If you don't give any argument to that one, it's going to select the first instance when there are two. But you need to do something. If you give that to the web server, it's going to return an error telling you have multiple side chains, and then you have to fix it. So these are all the things that you have to deal with. So these are the commands that we're going to run. So this is just illustrating the commands. So we have PDB fetch gets the PDB from the database. We clean it, we select the heavy chain of the antibody, we remove the heteroatom, we fix the insertion in a loop, we fix the double occupancies, we keep only the coordinate records, and we select only the first 120 residue in this case, which is the FV domain where the binding takes place. There is no need to model the full antibody, which is, two heavy chains, two light chains, Y-shaped molecule. If you can simplify your system, your docking is going to be faster. That's what we are doing. And we are writing that to chain H. And then you do the same for, we do the same for the second molecule. Now we are fetching chain L, okay? And then the third command will be merging those two chains, renumbering everything starting from 1 in this case. But there are also ways in PDB tools where you can shift the numbering. But anyway, you want a unique numbering, and for this tutorial, it's a unique, and we rename it to chain A. So the next cell of your notebook is going to do that. And you see that the files are appearing here in the directories. So the last one then, so once it's updated, the content here, you have the clean file, and the clean file contains basically the antibody with only the FV domain, renumbered, single chain. The last one here. So merge is merging two PDB into one. Okay, that's PDB merge. It's merge H and light chain. So these two files are the one that coming out of these two commands. Okay, and then I pipe it in Linux to the next command. I renumber everything starting from 1, so that you have a sequential numbering of the entire thing. I define chain as being A, and the next command copy the chain ID to the seg ID, and the seg ID, if you open a PDB file, the seg ID is found all the way at the end. I clean it to make sure that I have a proper PDB format, and I write it to this file. Have you all seen the inside of a PDB file before? No? Because you have seen it maybe in a graphical, you look at a protein structure, but it's a simple text ASCII file. Let's open it. This is the inside of a PDB file now. Okay? If you have never seen one, it's a text file. You have this atom statement. This is a atom number, atom name, amino acid number, the chain A, the residue number. Then you have the coordinates. Then you have a value which states the occupancy. Here everything is 1. So if you have two side chains that are 50-50 orientation, you will see 0.5, 0.5, and you will see duplication. This column has the B factor, which tells you how well defined is the position of the atom in the crystal structure. If you have AlphaFold model, the confidence, the PLDDT will be here. That's the seg ID. That's another information which is the same as this one in this case, and then you have the atom type at the end. Okay, so that's a text file. So you can open these and you can edit those files if you need it. But PDB, all the PDB tools commands that we've been giving, I've been manipulating those files. So for the antigen, we do the same thing. So the antibody was there. The antigen actually is it's interleukin B, 1B, I think. So we have crystal structure of the antibody in a free state. We have a crystal structure of the antibody on a bond state also, and we have a structure of the antigen. So now we have our both structure. We are ready to go. So we need to define restraints between those, okay? And in HADDOCK, a restraint is defined. I didn't explain that in my talk, but if you know that an interface is important, and you have two interfaces, how do you bring those together? So we don't use classical distance in a sense that the distance is defined between 2 points. That will be the Cartesian distance, the classical one, but we define ambiguous restraints, where we define a first selection. And the selection could be one amino acid, meaning all the atoms of that amino acid. The second selection could be a collection of amino acids on the other side. And then the distance that would be calculated will be based on the combination of all the possible distance that you can calculate. So all the pairwise combination of atoms between what is selected here and what is selected here will be defined. So you can say, you could even, if you were to design all the distance between the two proteins, it will be a bad idea because it will give you an n-square problem. You will say chain A and chain B in the syntax that we are using. The computational engine behind HADDOCK is called CNS. It stands for Crystallography and NMR System. It's a software which was devised to do structure calculations for crystal structure and NMR. But it has a very powerful syntax. Okay, if you want to define something similar in Chromax, you have to define all pairs, the indices of all the pairs of distances. And then there are 3 distances, the target distance, a lower limit correction, and an upper limit correction. So this is how those distances are defined. So for example, this one here could represent a cross-link obtained from mass spectrometry data, which will tell you that the distance is applied to the C-beta carbon of residue 10 of chain A and the C-beta carbon of residue number 200 of chain B. Okay, so you can have specific distance, you can have ambiguous distance. If you remove and name C-beta, the selection will select all the atoms of the residue automatically. And the distance, so the target is 20, the lower limit correction is 10, so 20 minus 10 gives you what is the minimal distance, 10, and the upper limit is 20 plus 0, 20. So this will define a range between 10 and 20 angstrom. So that's the way that, and you have a lot of flexibility in defining those. So we define the paratope of the antibody, and we use for that an AI model, which given the sequence gives you the most likely interacting residues, and those have been now renumbered based on the sequence numbering that we have in the file that we prepared. It's important if you extract information from somewhere that the residue numbering matches the numbering in the models that you are using to do the docking. If you have a mismatch in numbering, you are assigning information to the wrong residues. And here's the list of residues that have been identified, so we don't need to recalculate those. So we can visualize this directly on the structure. So if we are to doing the command line instructions from the Bonvin lab website, it will tell you how to visualize this in PyMol. So now we use Py3D-mol in the Jupyter notebook, and you can rotate your molecule, and you see here, this is the antibody that we are modeling, so it's only a small part of the antibody. And you see all the red regions are the regions that are predicted, and these are all the regions in the 6th loop of the antibody. So that's the information that we're going to give to HADDOCK. So this region will have to bind to the other region on the antigen. Now you could reuse this notebook and adapt it to your own system if you change the PDBs that you are working on. Okay, so you can play with those. For the antigen, in this case, there is an old NMR study from 2013, which actually also report the structure of this complex, so we know the answer to this modeling that we are doing. And in this paper, there is a table, table 5, which gives you which residues are affected by the binding, and this was identified by NMR. So they did NMR experiments that are telling you these are the residues that are sensitive to the binding of the antibody. So some NMR signal changes. And they did that for 2 antibodies. So these are therapeutic antibodies, you can see from the name. But we are looking at Jevokizumab. Anything ending with mab in names is an antibody. And the green ones are the ones that are affected by binding, basically. So this is the information that we have. So on the antibody side, we take the loop, basically. On the epitope side, we have NMR data. And this is the list of residue, and we can visualize so this is no longer an antibody, so this is a interleukin 1b, and you can see again that the NMR information is giving you one binding site. What you don't know is what is the pairing, okay? We don't have a specific distance, but we have one binding site on the antibody, we have one binding site on the antigen. And in case this was not working, this will be what you get if you do PyMol visualization. Okay. Now, one thing that that we are typically doing in HADDOCK, and the server will do that automatically for you, the information that we get from such experiments is not always very accurate. Okay, there are 2 problems. You might not be seeing the full interface because maybe some residues are not sensitive to the binding for some reason. And you might be seeing too much as well. Maybe you are seeing indirect effects like a conformational changes. An allosteric change might be seen by NMR, but it might not have to do something with the binding directly. So the way we deal with that is 2 ways. For the potentially missing information, what we do is to select the surface neighbors of those guys. And we're going to define those as passive residues in HADDOCK definition. And passive means that at the end of the modeling, if they are not in the interface, it's not penalizing you. It's not giving you an energy penalty. The one that we define as active, like these red ones, if they are not making contact at the end, you will pay an energetic price. Okay, so that's a distinction between the two. So that's solving we don't have the full interface, so we add the surface neighbors as passive. And the other problem is we might have too much information, we might have wrong information. And what's happening by default in HADDOCK is that for each model that we generate, by default, we're going to randomly delete 50% of the information. And we repeat this random removal of the data for each model that we generate. So from time to time, you're going to generate, to remove good data, and you might get wrong model. But from time to time, you might remove bad data, and you get better model. That's the theory, basically. And then you hope that your scoring function will be able to distinguish between those. If you know that your data are fully reliable, you can turn off. So that's a parameter in the setup of the docking. Okay. So again, this does generate a rather well-defined patch. Now, if you are unlucky, you see something here, and then you see something on the backside of your protein. Then you have a problem, because maybe there are two binding sites, or maybe there is an allosteric effect. So then you will have to think about how to use this information. One way, if you see two binding sites, will be to define those as passive, but not as active. So it could bind on one side or the other, and then you let the docking decide what is better. So this is defining the surface neighbors as passive. So there is a website for generating restraints that can do that, but we also have a command line interface in HADDOCK3, which is HADDOCK3 restraints. And this is going to find the surface neighbor. So it needs a coordinate files to do that, and a list of active residues, and then it returns back the list of passive residues. So this is part of HADDOCK. So if I run this, it calculates the solvent accessible surface area of the protein, and based on these, you get this list of residues that are defined. These are the surface neighbors. And we can visualize what this is giving us, and you see that things are here. So we have selected the surface neighbors. Well, some of those, I'm wondering if they should have been selected, because like this guy here is rather buried. It could be that the beta sheet is solvent accessible, but the side chain is buried, but it doesn't matter. So this is the information that we have at hand. Now, based on this list of residue, we're going to define our restraints. And then there are different ways. So there's a web server where you can do that. If I go to our web portal, which is called wnmr.science.unl, if you go under services, you will find a HADDOCK restraint server, and there you can input those lists, and then it generates the restraints for you. So now we're doing it here at the command line using HADDOCK3. So we're giving two lists of residues, a list of active for the antibody and no passive, and for the antigen of active and passive, and then it's generating a restraint file. And the restraint file looks something like that. See, the file has been generated here, by the way. So you see now a selection of distances, of ambiguous distances. So it starts, so we have this assign statement, so that's one restraint. The first one tells you residue 31 and chain A. That's the first selection. And then we open the parentheses for the second selection. And then you have a long list of OR statements. So is this residue, or this one, or this one, or this one? So it's a long list. And here they are, basically the list contains all the active and passive of the second molecule. So you see that there are probably more than 30 amino acids that are selected here. Let's say it's 40 amino acids. How many atoms on average in one amino acid? Any idea? How large are the amino acids in terms of number of atoms? Forget about hydrogens, okay? So we think of amino acid. So how large is an amino acid? Is it 5? Is it 10? Is it 100? 10. On average, 10 is reasonable. Okay, we have 4 on the backbone, C alpha, CO, and the nitrogen, and then the side chain. And some are very short, some are very long. So say on average 10 amino acids. So here on our long list of selection, for the second part, we have 40. So this is 400 atoms that we select that we have to combine with the first selection, which will be again 10, say. So the number of combination is 10 times 400. So there are 4,000 individual distances that are used to calculate one effective distance. If there was a board, I would write what the distance is, but it's basically what we calculate. So we calculate all those individual distances. We take the sum of 1 of the distance to the 6th power. Have you seen 1 over r to the 6th power this week? Maybe in the molecular dynamics part. I don't know. Have someone shown you the equation for the van der Waals energy? 1 over r to the 12 minus 1 over r to the 6, something like that. So that's the attractive part of a van der Waals Lennard-Jones potential. But 1 over r to the 6 is also dipole-dipole interactions, if you are thinking NMR. And so we sum all the distances, so 4,000 distances, we sum as 1 over r to the 6. Then we take the inverse of that sum and the 6th root of that sum, and that gives you 1 distance out of that. Okay, so we have 1 effective distance, which is calculated from the combination of those 4,000. This is what the distance that we use to define the energy of the system. Okay, and it means that if 1 of the 4,000 distance is short enough, our restraints will be satisfied. It doesn't matter which one, as long as 1 is short enough. And this is basically allowing you to have docking solutions that might be like this or like this. Okay, so it does not define the orientation. So it has freedom in orientation. You just want that they should be contact. So we have this network of ambiguous distance between the molecule. And for each residue that you define as being important for the binding, you will have 1 such distance calculated from all the combinations. So you can see so we have a network of ambiguous distances that are pulling the molecule together when we do a docking. And you can encode many types of information in these ambiguous restraints. Okay. So if you write manually those restraints, there is a command to validate to make sure that you didn't make any mistake, and it's just checking that the syntax is correct. You know, if you open a parenthesis, you should close it, things like that. So here it doesn't report anything, so everything is right. Another small thing that we have to do here has to do with antibodies, because the antibody is 2 chains, and the domains that we're looking at, they are not covalently attached, so there are no chemical bonds between the 2 domains. So when we're going to do a simulated annealing that I explained in lecture, you know, we start high temperature, we cool down the system, we do molecular dynamics. If the chains are not attached to each other, they could dissociate, or they could do bad things, which are going to affect your structure. This could also happen if you have a single protein, but then you have a loop that has been cut, and that loop allows the system to separate in 2 parts, which can happen if you use the AlphaFold model and you remove the spaghetti. So we want to define restraints that keep the parts together. When you use the web server, this is done automatically. You don't see it. The server checks for you. But here we are using command line, so we need to do that. And that's what this HADDOCK restraint bodies does. It checks your PDB. If it finds breaks in the chain, it's going to define a couple of distances just to keep those things together. So that's this one. And now basically it randomly picks 2 distances. So now the distance are reading between 2 atoms, so that's a single distance. And it has measured the distance from the PDB file. So, and you see that the error bonds are 0. So you just want to keep the things together. Okay, so we have the PDB files, we have the restraints who are ready to do the docking. We need to define our workflow. And this is the workflow that we're going to run. So I told you we need to start with the topology, so that's the first module. Then we do our rigid body energy minimization, the first step in HADDOCK 2, rigid body. We're going to calculate some Capri metrics after that just to know how good the models were. We're going to select a number of models that we pass to the flexible refinement. Flexible refinement takes a bit more time. Rigid body docking, it's seconds. Flexible refinement might be tenths of seconds or a few minutes, depending on your system, per model. We again analyze some metrics. We do a final refinement, energy minimization. We do clustering based on contact. We select the best model of the clusters. We do again some analysis just to get the metrics. Each analysis module, Capri Eval, will generate this interactive table and plots, so that's nice to visualize your data. And at the end, we analyze the intermolecular contact between the molecule. So this is the sequence of the workflow, and the workflow is defined here. So we define our run 1, or whatever name you want. The compute mode of HADDOCK is local, meaning it's going to run on the system that you are. It's not going to, there are different compute modes. We have local, we have MPI. So if you were to submit to your batch system in MPI mode, you could ask for multiple nodes, and then you say, I'm going to distribute. If I calculate 200 models, you might want to have 4 nodes with 50 cores, and then you could run on more than one node in that way. And there is a batch mode that's sending single jobs to the batch system. That's not usually the running mode that they like in HPC system, because you are sending a lot of small jobs. So on Fugaku, when you open an interactive session and say, give me the full node, by running in local, you're going to fill up that node with HADDOCK computation, and each model will be a separate calculation, okay? That's also the mode that you run on your laptop, typically. These are our input molecule that we just created, and then you see here the different modules, but we need to define the restraints file that we're going to use, and we have 2 restraint files. The ambiguous file, which is defined from the paratope and the NMR data, and the file to keep the antibody together. We limit our sampling, so we're only calculating 100 model. We have good data, so we don't need to sample that much, and for the speed of the tutorial, we select the best 20 model after the rigid body docking, which we give to the flexible refinement. The tolerance is a percentage of model that are allowed to fail. It could be that during the simulated annealing something crashes for some reason. So if we have 5% failure, we are okay. It will keep going. If there's more than that, it will stop. Again, some assessment. Minimization, again, with the same set of restraints. And then we see that in a Capri eval, we're using a file which is provided together with the data that we downloaded, which is basically the reference structure of that complex, a crystal structure of which the numbering has been matched to the numbering that we have now. In principle, this is not really needed in HADDOCK 3 because the Capri eval module will do a sequence alignment and try to match the structure also and match the chain. So this is a workflow. And then we are ready to run HADDOCK. Okay, so there are 2 ways that you could do that in this notebook. If you want to run, anyone running it already? Or maybe it has finished already while I was speaking, yes. So if you want to run it interactively in a workflow, you run that one. If you manage to get the CPU on Google, that was taking 11 minutes when I tested this morning, so it's quite fast. If you don't want to wait, you could open a terminal session and then send it to the background and it should run. So in my case, since I think I only have 4 CPUs, which means it should take about 4 times 11 minutes to run. So I'm going to copy this. I'm going to go into the terminal. Oh, I don't need to do, it's running already. HADDOCK is starting. It's checking the validity of the workflow when it starts. So if you have made a mistake in a parameter name, it's going to tell you that. It's not going to overwrite an existing directory. So if you run the same run, it's going to stop because it's telling you this is already existing. So then you have to either remove it or do something about it. And now you see that run 1 has just been created. missing atom would have been added. And now it's doing the rigid body. And if I look into run one rigid body, you see that some more will start to appear. Okay. But I've limited CPU, so it's going to take some time. Let's move it away. And you can also see things appearing here in your window if you open that one. So that one is running for me. If for some reason, you know, something goes wrong, or you were running things and then you were kicked out of the node because you exceeded the time that you were asked for, you don't need to restart from scratch. If you say the first 3 stages completed fine, but I need to start from 4 again, there is an option that you give in the command line minus restart 4. It's going to start at the 4th stage of your workflow. If one stage fails, at this time you want to generate 1,000 model and you've got 800 and you just need 200, too bad, you will have to run that entire stage again. So we don't have a restart mechanism where you will see what is done already.